Bismillahir Rahmanir Rahim. Dear students, Assalamu alaikum and welcome back to our cell biology, genetics and evolution class. We were discussing genetic engineering, and in the previous video we discussed restriction enzymes. And now we are going to take a look at the very important enzyme or molecule which is used in genetic engineering, and that is DNA ligase. We have already studied this molecule or the function of its molecule when we were discussing DNA replication. That when DNA polymerase 3, especially in the lagging strand, when it completes the synthesis of a small segment of DNA, and then it continues to do that, and those segments are known as Okazaki fragments. So this is the template strand and these are the Okazaki fragments. So the final nick that remains after the primer is removed, and the space is filled by DNA polymerase 1. So DNA ligase then comes into action and it seals that final, what we call the nick, the final phosphodiester bond that is broken or it needs to be made, synthesized. So DNA ligase does the job there. And in this particular case, as we are going to see that what DNA ligase plays a part in genetic engineering, we have already seen that gluing or connecting or joining various DNA molecules. So what it does, that it repairs that phosphodiester bridge. For example, we have two nucleotides. This is one nucleotide with a phosphate at this end, and then this is three end, and for example this is another nucleotide, and it's now if this particular bond is broken, so this is the job of the DNA, which it does very efficiently in DNA replication and also in genetic engineering, that it forms or creates this particular bond. So these nucleotides will be joined together, and this is the nick that I was just talking about. So DNA ligase creates or synthesizes or forms this particular bond. As I just told you, that DNA ligase acts like a glue, joining the DNA molecules when DNA molecules are cut, and when it's needed that those molecules may be joined, so DNA ligase comes into action. And in genetic engineering, it is used to, as it says, seal the discontinuities in sugar phosphate chains, that is those phosphodiester bridges that are not there or absent. So it creates that bridge or seals the discontinuities and joins the DNA molecules together. And how do these discontinuities arise? Obviously, when we break the DNA molecule into fragments or when we break the vector molecule, so obviously those bonds are broken. So that is these discontinuities arise when recombinant DNA molecule is made by joining DNA molecule from different sources. So when we want to join them, so obviously there are discontinuities, and those discontinuities are sealed by or removed by the DNA ligase. So molecular glue, again, stick, join, unite, whatever you call it, combine pieces of DNA together. Obviously its function is very crucial to the success of obviously this genetic engineering experiment, and it's a key enzyme in genetic engineering. Now, where do we get this particular enzyme? How do we get it? DNA ligase. So this is the T4 DNA ligase which is used, and it is obtained from E. coli cells, especially those cells which are infected by T4 bacteriophage. So from those cells we purify or obtain this particular enzyme, the T4 DNA ligase. So that was a discussion of DNA ligase. We didn't see much of it, no example, as I told you earlier in the beginning that it's a very basic introduction to genetic engineering. So we are not going to go into more details. Since most of the job of genetic engineering is done in the lab or in vitro, that is creating fragments of DNA and then joining them together and joining them with a vector molecule. So that's simple. We do it in the lab. But what we need next, what we have created, the new DNA molecule or the recombinant DNA molecule that has been generated, we want to amplify that or we want to use it. So obviously we need some mechanism to make use of that particular recombinant DNA molecule. Therefore, once a DNA molecule has been made, it needs to be amplified. That is, we need more copies of it so that we can carry out more experiments or manipulate it further. So for that, in genetic engineering, we need a suitable vector molecule, a carrier molecule that would carry our desired DNA molecule, the recombinant DNA molecule. And most often we use plasmids as vector, but we also use viruses as we saw in the previous video. And then we need a host in which this vector can be propagated. That is, we need to amplify, we need to create more copies of it, or to make that DNA express itself if we want to create or make a product. So we need a living host, a system, what is usually called host in genetic engineering. And obviously, there is a long list of hosts that are used in genetic engineering, but the usual host is bacteria. So for the time being, we stick to this simple discussion. So these were the various tools used in genetic engineering: the restriction enzymes, the DNA ligase, the vector, and the host. So now we are going to see in the next couple of slides an example of genetic engineering. So this is a human cell. We take human DNA from that particular cell and we have isolated insulin gene because we want to create insulin, for example. Then we take a plasmid from a bacterium. The plasmid is cut open. We make use of the restriction enzymes, which obviously remove fragment of the insulin gene from human DNA and simply cut open the plasmid molecule. So when the plasmid molecule is cut open and we have these insulin genes, next is the function of DNA ligase to join these two molecules to create a recombinant DNA molecule. So now the insulin gene is incorporated into the vector and this is a recombinant DNA. Now we have introduced this particular into a host cell. So this is a vector which we were just talking about, and this is the host. This bacterium is the host. Now what happens that when bacteria multiplies, it creates copies of those genes. So all these bacteria, the daughter or the progeny, they are carrying that particular gene, and when this gene is given a living environment, suitable environment, then this insulin gene would express itself, and what would we get in the end is the insulin. So this is a very basic and simple example of genetic engineering. In this particular slide, as you can see, we have some uses of genetic engineering. And this is if we want to do a basic research on gene structure and function. What is the structure of a gene and how it functions? Then we want to create useful proteins, as we have just seen production of insulin, which is also a protein. By novel methods, obviously, we can craft or create our own methods in genetic engineering and we can create useful proteins. We can create transgenic plants and animals, and this is a practice which is going on now, what we call them GMOs, genetically modified organisms. what we do that we introduce foreign DNA into them, and when that DNA is expressed, obviously that is DNA is of our choice, and it has some because of that DNA we want to create a required or a suitable trait or phenotype or a character in that particular plant or animal, as I gave you an example in the very beginning of poultry. And similarly in plants, for example, we want to have or create a wheat plant which produces more grains and big and healthy grains, for example, so better yield and better grains. So transgenic means plants or animals in which a foreign DNA has been introduced. So generation of transgenic plants and animals. Obviously in medical diagnosis and treatment, that if a disease is because of the genetic material or DNA, so we can also diagnose it and suggest treatment. And similarly, we can analyze the genome, the DNA of an organism by sequencing, and all these tools are used like restriction enzymes and DNA ligase and amplification and so forth. So these are just a few simple uses of genetic engineering, but we can do more than that. And obviously this you will study this in your coming semesters. So this concludes our very introductory and basic discussion of genetic engineering. Thanks for watching, and see you in the next video.